Okay, genome and AI-informed individualized care of infants in intensive care units. What I'm going to talk about in 20 minutes is the single most significant opportunity for changing outcomes in human health care by individualized medicine in the next 3 years. Let's see if I can substantiate that comment. That's self-explanatory. Okay, so this cartoon shows the standard of care, not individualized medicine, standard medicine for management of infants, that's children less than a year of age, in intensive care units in the developed world. So 55% of children admitted to an intensive care unit have a disease of unknown etiology. They have a presentation, they are empirically treated for the presentation, the etiology is unclear. What occurs for those children is concomitant with interim empiric therapy. There's a full court press, good March Madness term, to determine what is that etiology with lots of test ordering. The children get better or worse, and treatment is modified as the differential diagnosis is refined, as results come back, and as they respond or fail to respond to the empiric management. This doesn't work well for children with genetic diseases. So our studies have shown that 15% of infants in intensive care units, that's neonatal or pediatric intensive care units, have a genetic disease. And this paradigm of standard of care medicine doesn't work for these children because there are 14,000 genetic diseases and they are inevitably being misdiagnosed and mistreated. In fact, 60% of them will go undiagnosed during their stay in an ICU, and that leads to mistreatment and adverse outcomes. And in fact, genetic diseases are the leading cause of infant death in North America as a result of this failed paradigm. So my talk's going to be back to the future. So first of all, let's go back and look at the evolution of individualized care for children in ICUs. So the first genome we all know about, 13 years. The first medical genome followed 6 years thereafter with the advent of Illumina next generation sequencing. Took 1.5 years. 2012 was my previous appearance here when we had just set a record for being able to telescope that time down to 50 hours. So 50 hours from request for a genome to making a provisional diagnosis. At the time, that seemed pretty impressive, but it didn't really work in the real world. This was a protocol and experimental tool. 2015, we had the first clear evidence that this affected outcomes in infants. And furthermore, we had a new version of this test, which shelved the time further down to 26 hours. 2018, last year, we set a new Guinness World Record, which I'll show you, for fastest medical diagnosis of median of 20 hours, shortest time 19 hours, and published some of the first effectiveness studies. Let's take a look at those two studies. So first of all, this was the first efficacy study. This is the point in time when my life changed as a research scientist, when we demonstrated, much to our surprise, that individualized medicine was a generalizable phenomenon in infants in intensive care units, and that the bottleneck hitherto had been the lack of a diagnosis. And not only did we find that we were able to implement individualized medicine in 31%, but that it changed outcomes in 11% and saved 1 life. And when we compared the speed of this with the standard prior tests, including next generation tests, you can see that not only did we benefit in terms of improved rate of diagnosis, but the speed was really an important attribute in terms of implementation of individualized medicine. That was 2015, and the methods then were primitive. If we skip forward to 2018, a more recent examination, different ethnic group, different ICU, different methodology, but the same intent, and the same result, that we have changes in management in 31% of infants and 26% have a change in outcome. This has now been recapitulated in over a dozen studies with groups from around the world. And these days we have even faster methods of diagnosis, and you can see that again, for the most critically ill infants, it has a higher rate of diagnosis, and now we're finally down to a time frame that's meaningful for care of acutely ill infants. And so in 2019, today as we sit here, this is the new status quo, or state of the art, I might say. For the 55% of infants in intensive care units who have diseases of undetermined etiology, a rapid genome sequence, when bundled into that initial set of tests that are ordered, results in what I've just shown you in the prior slides, which is that we will have a provisional diagnosis within a day and a half minimum, and furthermore that we will give a genetic diagnosis in about a third. That means in 2/3 we rule out, by and large, a genetic disease. That is phenomenally useful in terms of implementing genomic medicine. About 25% will receive that, and 20% will have an improvement in outcome. So very simple numbers to remember. 1 in 3 get a diagnosis, 1 in 4 receive individualized therapy, 1 in 5 have an improvement in outcome. So that's great, the job's done, right? Well, not really, because the reality is that today in 2019, less than 1% of infants in US ICUs will actually receive individualized medicine in the way I've shown you, and the 99%, it's like a biblical parable, right? The 99% get the old non-individualized method of care, which means for the 15% who have genetic diseases, they're still suffering. They're still being misdiagnosed, mistreated, and having unnecessarily poor outcomes. So what are the barriers to go from 1% or less than 1% to a majority adoption? This is really what we're focusing on heavily now. And there are 4 of these. I'm going to focus mainly in the next couple of minutes on the top one, which is that the methods previously described for making an ultra-rapid diagnosis had an immense capital and labor intensity, something that for most children's hospitals was unrealistic to deploy. And so we focused heavily on that. I'll come back to the other barriers at the end of my talk. So we believe that we've made substantial progress in knocking that barrier down. That's me with a cheesy grin, and this was last February when our multidisciplinary team did manage not only to shorten the time to a diagnosis to 19 hours, but much more importantly, to make a diagnosis entirely devoid of human analysis or interpretation. So full automation from sequencing through return of result. Let's take a look at that. And let's do that in the setting of a real live case from this past year. 17-month-old boy is seen in Rady emergency department with fever, nausea, vomiting, diarrhea, abdominal pain, labored breathing, and very unusual skin lesions, which are shown here. We immediately plug him into the circle that I showed you, where he gets the full court press workup. And the relevant results are shown here. He has a metabolic acidosis. His inflammatory markers are grossly elevated. His white cell count is low, which is ominous. He has a workup for his belly pain. This is gray medicine. There's not a clear-cut diagnosis coming from that. He has an infectious disease workup, which includes blood cultures and also a lumbar puncture. And during that, he has an event where he acutely decompensates in terms of his hemodynamics and his respiratory status, and he requires fluid resuscitation and ventilatory support. He started on empiric broad-spectrum antibiotics for suspected septic shock or pre-shock and admitted to the intensive care unit. Hospital day 2, we have a diagnosis. He has pseudomonal sepsis. This is a very severe gram-negative bacterial pathogen associated with poor outcomes. Furthermore, we realize that his load of bacteria is very high, because the skin rash are actually little gangrenous lesions which are septic emboli as a result of his infection. This is worrisome not just for his skin, but also because he's having septic emboli in other major organ systems. So a rapid genome sequence is ordered. We've gone a long way since 2012. These days, we've optimized the order interface. It's in our EPIC EHR, and a genome is not quite a regular test. We need to think through to what extent do we want to report results. Do mum and dad want to know about their health? Do they want to know about the future health of their infant? Maybe yes, maybe no. We've got tick boxes. And then lastly, my favorite button, the one on the bottom, is it foot to the floor? Do we go for the world record again in this child? And for children like this one who have a high likelihood of morbidity or mortality, we say yes. The first step is to make a library, to get DNA out of the boy's cells and have that ready to put on a sequencing instrument. Typically, this is a multi-step process which takes a day. Using the Illumina Nextera kit, we can go straight from blood to library in 2.5 hours with many fewer manual steps. Second of all, we put this on the world's fastest sequencer in its fastest mode. That's the Illumina NovaSeq, introduced at the start of 2018 with the S1 flow cell. And 15.5 hours later, we will have 5 genomes completed at 40x coverage. So we've decoded the infant's genome 40 times. From here on in, everything is automated. The first thing is to take those raw sequences, 125 billion DNA letters, and turn that into the child's genome code. We do that using a Dragon system. That was a company from La Jolla, actually, called Edico, who were acquired by Illumina. And then from there, we want to identify all of the changes in that child's genome from a reference sequence, all of the variants which could be causing a genetic disease in this child. Those 2 steps, astonishingly, now take only 45 minutes without the need for a supercomputer. Next, we want to say which of these 4.9 million variants could be causing this child's illness. We do this in what we will look back on today as a primitive way, which is to use consensus guidelines that experts have assembled, which say whether a variant is likely to be disease causative or non-disease causative. They kind of work. More importantly, they can be turned into computer script, which allows us to compute these very quickly on all 700,000 variants. It's only 700,000 because we're able to eliminate those that are common, just based on their frequency in populations. And that gives us, within 2 minutes, about 1,000 things in the genome which might trigger this child's disease. Okay, what we do next is not obvious. We go back to the child's medical record. Suddenly, we are now dependent on the skill with which the physician took a medical history, did an examination, and the results of those initial tests. So artificial intelligence, when applied to healthcare, and individualized medicine don't replace the physician. In fact, they place a higher emphasis on the adequacy of those clinical skills in order to instruct the artificial intelligence engines. And infants are unique. They have a lot of stuff going on that is not related to a genetic disease. So on the pink box shows everything that's going on in a typical intensive care unit child based on a series of 76. So on average, 93 different phenotypes or clinical features. The blue box shows their disease description. Each of these children has a genetic disease. What you can see is that there's on average about 93, no, that's incorrect, there should be about 40 phenotypes in the textbook description, and the overlap is minimal. But in the green box is how we traditionally order genetic tests. We put in some simple descriptive terms like pseudomonal septic shock to describe our patient's illness and expect that we can then turn that into a diagnosis. Well, we can, but that takes expert interpretation. And when we move to an AI solution, we need a deep phenotype, or what we call a phenome. And furthermore, as you can see from this, we need to not penalize non-matches because the matches are slim. The child has not yet grown into the textbook description of his illness. So here's how we do that. First of all, we spend about a year optimizing natural language processing so that it can automatically extract the child's phenotypic features in a comprehensive and accurate manner from the medical record. It takes 20 seconds. Second, we want to translate those from text, from physician jargon. And physicians love their jargon and their abbreviations. Oh, dooey boy. We can think of an abbreviation for anything. We need to translate that into a machine-readable language. And the critical thing here is that any clinical feature has a certain amount of information content, and if we want to build a diagnostic engine, we need to weight terms according to their information content, according to the frequency of occurrence in the medical record, and we want to not penalize slightly imprecise matches. As you can see here, we will have certain terms which precisely match what the textbook says and others that are close neighbors. And we need mathematically to have a robust manner in which we can assimilate all of that information and compare our child, 17 months old, with 14,000 genetic diseases. So we do that, and a whole variety of tools have been built to allow us to generate almost instantly a comprehensive differential diagnosis. None of these fully embody everything that I said in the last 30 seconds, though. There's a lot more work here and hard math to be done to optimize this process for rare genetic disease. Nevertheless, we are able to do this, and it takes about 2 minutes to not only do that, and remember that every disease is associated with a gene, and every variant is associated with a gene. And so it's very simple matching to unify on the basis of genes and say, do any of the genes that match the differential diagnosis also match a variant that we've seen in this child? And the answer in this case is yes, and it's 1, and we have a diagnosis. The box shows a screenshot of the tool. It's made by a company called Diploid, a screenshot of the actual provisional diagnosis. Again, absolutely no human intervention from sequencer through this screen popping up. The child has X-linked agammaglobulinemia. That's a primary immune deficiency, and that's what his etiologic diagnosis is. That's why he has pseudomonal sepsis. And there are some criteria there that speak to the attributes of that particular variant that we don't need to get into. A critical one is this is a test result like any other test result. The clinician then needs to switch back on his brain, get out of AI mode, and determine, does this make clinical sense? Do I need confirmatory testing? We have that already in the chart. The child has hypogammaglobulinemia. He has a low B cell count in his blood. So the functional evidence supports the diagnosis. The happy ending is that the child immediately gets individualized medicine. The treatment is to supplement his immunoglobulin with intravenous immunoglobulin to change his antibiotic intensity and length and to continue to pursue the complications. He has a good outcome, and he's discharged on day of life 13. His prognosis is excellent. There are also family implications for mum's relatives, since this is an X-linked recessive disease, and this is another component of individualized medicine. It's not just about the patient, it's about the family. And the consequences in terms of health benefits for the family can be quite profound in terms of prevention of recurrence. So a beautiful case example of individualized medicine triggered by a rapid genome. Here are prospective patients, 7 of them, and in the darker blue is this case 7052. You can see that we made a diagnosis in 22 hours and 4 minutes. Our fastest standard methods using humans took 38 hours and 37 minutes, 16 hours of savings, but much more importantly, we've alleviated the labor and capital intensity of this test in a way that can be scaled to 40,000 infants who need this per year. Over 7 cases, we got 3 right and no false positives. When we retrospectively looked at 84 children, you can see that the precision and recall of the artificial intelligence solution resembled that of experts in terms of manual interpretation. So let me wrap up. We are at this barrier to adoption phase in terms of implementation of individualized medicine for infants in intensive care units across the civilized world. Some healthcare systems, like the British healthcare system, have said they are going to adopt this nationally. Now, that doesn't mean that they will just flip a switch and it will be adopted nationally. They will have these same barriers to adoption. But they have already made that at a societal or governmental level, they've made that decision. Here in the US, decision-making is a patchwork quilt of payors and states, and it's going to take longer. So what are our additional strategies to address these barriers? Well, first of all, we now have a method I really think we can scale to 40,000 individuals a year. It needs some work, but it's looking very, very good. But what are the other barriers? Well, a critical one is that we can deliver a test result and it not turn into individualized care because a clinician is not familiar with this concept. They don't know what to do with that data. They don't know which children to order a genome test in. They don't know how to give genetic counseling. And so what we want to do, just as we built the test order into the EPIC EHR system as an API, we want to do the same with all of the missing pieces. Can we provide 90% of genetic counseling via frequently asked questions for that condition? And can those be available through an API? Can the management guidance be also available? Now, there's a lot of work to be done. This knowledge base has not yet been organized. Much of the knowledge base of the management of rare and ultra-rare genetic diseases is in PubMed. It's not in a single location. It's not been standardized or vetted. And so we need to do a lot of work to get that also into a structured format so that just as we can make a diagnosis very rapidly, we can immediately go to optimized treatment without having to dig into the literature, pull it, and read through manuscripts. And then lastly, we need more studies that are attuned to the types of data that payors need so that we get the same decisions in terms of prior authorization and reimbursement. And so let's end up with Back to the Future and say, where do we think we want to be in 2021? Well, we would love to see that we are in that early majority where 25% of children's hospitals will be doing this for a substantial minority of the children who have a disease of unknown etiology. And what this means is that we're changing from research mode into implementation science mode and quality improvement mode, where we really address what are the practical barriers in a working intensive care unit to deployment of this and to having it result in better outcomes. Second of all, there is no second of all. First mistake. Almost perfect, Kingsmore. Sorry, Eric. What can we expect by 2021? That we should be able to generalize what I've shown you. 1 in 3 will be diagnosed, and very importantly, in the other 2/3, we have changed the posterior probability that they have a genetic disease, also phenomenally important in decision-making in an acutely ill infant. 1 in 4 will have individualized medicine, 1 in 5 will have an improved outcome. 3 years or 5 years further on, those numbers are going to get substantially better because, as I've said, we haven't yet attacked the knowledge base, and only by doing numbers will we see improvements in terms of individualized medicine's efficacy and outcomes. So thank you very much. I have a saying which is ad deo lumen ab amicis auxilium. which speaks to the fact that this is highly multidisciplinary work and requires the creativity of a team, not an individual. I get to speak, and these are the folk who contributed to this and are deploying and implementing this in currently 16 children's hospitals around North America. One other very quick note, you can see that I've acknowledged a number of biotechnology companies. And another part of individualized medicine is this is not going to happen as a pure play academic process. It requires true bringing technology to the bedside that requires innovative partnerships where we are able to work closely with commercial partners and their scientific rigor to enable breakthroughs. Thank you very much.